Alright, Assalamualaikum, selamat pagi semua. Okay, so today we do experiment number 2 and 3. Okay, so number 2 experiment for unicellular fungi, which is yeast, and experiment number 3 will be filmentous. Okay, fungi, which is we using what we call mold. Right. So please be ready with your notes, and we are going to brief this experiment as well. Thank you very much. All right, so the first experiment number 2, so we are going to do the unicellular fungi, right? Okay, so experiment number 2, unicellular fungi. Example is what we call using yeast, all right? So there will be introduction for the experiment. So the fungi kingdom contain a large group of organisms, okay? The range in size and complexity from one cell yeast to filamentous mold and to the complex mushroom. So mushroom, yeast, and mold will be one of the example of fungi. Okay. The one cell yeast reproduce asexual of by what we call budding. Okay. So a budding daughter cells begin as a small protrusions of the mother cells. Okay. So dia akan mula cambah daripada mother cells. Okay. Since this is a mitotic type of divisions, which mean mitotics mostly process of the same species, same sample. Kalau macam dia punya host mother dia bulat, anak dia bukan dapat bulat, tidak beza, alright. So lebih kepada mitosis, alright. An equal amount of genetic material is given up from the mother cells to the budding daughter cells. Okay. Therefore, the inheritable characters of the daughter cells are the same, okay, as those of the mother cells. Although the daughter cell is usually smaller, indicating that there is not an equal dimension of cytoplasm, which is true and seen in bacterial fissions. Okay, so bacterial fission, binary fusion semua sama, alright. 2 pada 4, 4 pada 16 dan sebagainya. Okay, right, continue. So yeast is unlike the mold. Okay, why? Because they look more like a bacteria. Okay, so yeast adalah jenis mold, tapi dia sebenarnya ada kekurangan fungi, tapi dia lebih kepada bentuk dia seperti bakteria. Okay, when grow in artificial media. Okay, tapi dia masih lagi dalam bentuk fungi. This can be simplified observation from the yeast trick plate after incubations. Okay, so once awak tahu tentang bakteria, fungi, so macam mana, kenapa dia sama? Sebab bekas pertumbuhan dia macam biconscave juga, macam bakteria kalau awak pakai yeast, alright. So a few of yeast are medically important. 2 examples that can given in this experiment is Candida albicans and Cryptococcus neoformans. Okay, so what is Candida albicans? It's a normal inhabitant of the intestinal tracts, but it's one of the opportunistic pathogen. Let's say kalau kita ada masalah sikit je, sakit perut dan sebagainya, so these types of yeast akan jalankan proses pathogenik. So cara kita nak bunuh dia gunakan antibiotic treatments, but in this case we have to use it prolong, okay, lama makanya sekejap. So the observation of pseudohyphae and germs tubes are diagnostic. So walaupun kita katakan yeast tu macam lebih kurang bakteria, tapi yeast ada juga ciri-ciri yang ada pada fungi. Contohnya adalah pseudohyphae. So bila kita tengok dalam eksperimen, kalau tengok dalam gambar-gambar yeast dan sebagainya, mesti ada pseudohyphae. So that's why kita kelaskan dia sebagai fungi. So yang kedua adalah Cryptococcus neoformans. It causes fatal meningitis in human and produced in large capsules. So meningitis ni letak pada kita punya otak. Okay, meningitis. Alright. So kesan dia mungkin encephalitis ataupun kepala besar. Okay. The diagnosis is by doing a negative stain on the spinal fluid of the patient. So kita pakai cecair tulang belakang. Right. Meningitis ni letak pada otak kita. Right. So for today's activities, number 1, to revise yeast streaking methods. Okay. So this one you have a video. So boleh tengok video tu dalam saya dah masukkan dalam YouTube. So boleh tengok macam mana kita nak streaking method sebut. Yang kedua, to compare the appearance of unicellular fungi, yeast and bacteria. Number 3, to observe the morphological differentiations of yeast cells. Okay. Okay, what is material yang kita perlukan ni? Okay. Number 1, you must have what we call equipments. So what type of equipments? Water bath. Microscope, okay, because we want to see the morphology part. Microfuge tubes, glass slide, and glass cover slip. What type of stain kita gunakan? Lactophenol cotton blue or LPCB, or a simple stain iaitu sebagai methylene blue. And then Indian ink. Okay. So for yeast, we're using what we call media, what we call saburut agar or serum. Okay. Alright, what type of culture yang kita gunakan? Candida albicans, okay, Saccharomyces cerevisiae, Cryptococcus neoformans. Last one, to compare between all these fungi, we're using Bacillus subtilis. So yang dalam kurungan ni adalah dia punya media, okay, okay. So kalau Bacillus subtilis dalam kurungan ni Na adalah nutrient agar. So untuk Candida albicans, accept Zepec dog plus twin 880, right? Ini adalah dia punya media yang kita culturekan kita punya fungi tadi, right? Okay, number 1 method is a dilution streak. First you have to flame the loops. Okay, loops macam bakteria juga. Slightly touch the yeast colony with the flame loops. Alternately, yeast suspension can be used as an inoculum. Okay, ambil sedikit kat atas tu. Lepas tu awak streak pada set plate. Okay, streaking sama macam streaking bakteria tersebut. And then incubate at 30 degrees Celsius overnight. Okay. So B, comparisons between yeast and bacteria cells. Okay, number 1, using a flame wire loops, remove a small portions of the yeast or bacteria colony and mount in a drop of LPCB or methylene blue on the clean glass slide. Cetak titik atas awak punya glass slide. Ambil sedikit portion macam sistem bakteria juga. Okay, continue. Place a cover slip on and examine under the microscope. Okay. You might need to observe bacterial cells under higher power than the yeast cells. So, dalam bakteria kita pakai mungkin dalam 100 kali magnification rather than we use it for yeast. Sebab yeast besar sikit daripada bakteria. Okay. And then make a sketch of few cells, make notes wherever appropriate, for example, size or arrangement of the cell. So this one is more on morphological part, the samples of bacteria. Okay. So this one is more on the macro part. Okay. What about microscopic observations? So microscopic observation of the yeast differentiations. Okay, number 1, we're using what we call yeast pertama, iaitu Candida albicans and Saccharomyces cerevisiae. Alright. So pertama adalah kita kena cari formation of chlamydospore ataupun pseudohyphae pada dua jenis yeast yang kita gunakan, iaitu Candida albicans dan juga Saccharomyces cerevisiae. Okay, this is example of yeast. So yeast ada dua spesies, iaitu Candida albicans dan juga Candida albicans and Saccharomyces cerevisiae. Right? So how the work's done? Add 1 ml of distilled sterile water to a glass tube and make a suspension by taking half a loop full of each yeast. Okay. And then place a loop full of a suspension on a small petri dish or Zafek dog plus twin 80 medium. So Zafek dog plus twin 80 ni dalam bekas dalam petri dish tu ada dia punya medium tersebut. So awak pakaikan, dia macam bakteria awak ambil loop, lepas tu awak dia strip je pada awak punya media ni. Okay. And then you have to place a sterile cover slip on the top. Okay. And then incubate at 27 degrees Celsius for 24 hours. Alright. Examine under microscope at the lower power. Okay. So cara dia, awak ambil cover slip, awak ambil slide, letak sedikit air atas tu, okay. Lepas tu ambil loop of suspension yang kita tumbuhkan kita punya Candida albicans dan juga Saccharomyces cerevisiae tu dekat dalam plate tu. So kita ambil loop tadi tu, kita slide je atas kita punya sampel tu. Lepas tu kita letak dalam cecair air yang kita letak dalam slide tu. Lepas tu awak letak cover slip. So konsep dia sama macam bakteria juga. Okay, ambil kita punya koloni sedikit. Okay, letak sedikit air atas kita punya slide. Lepas tu kita ambil koloni dalam loop tu. Lepas tu kita color, kita letak atas cecair tu. Sterile water tu. Lepas tu kita letak cover slip. Sama je konsep dia. Okay. And then germ tube test. The test is performed to identify Candida albicans. Yeast Saccharomyces cerevisiae is used as a negative control. Place a small portion of colony of each species in 0.5 ml of serum. Incubate 37 degrees of Celsius for 2 hours. Remove a drop of the suspension from the middle of the tube onto the clean glass tube and place cover slip on top of it. Okay, sama juga ambil portion koloni tu ke dalam kita punya serum tadi. Okay, serum tube germ tube kita pakai germ tube, so dia dalam bentuk serum. Lepas tu kita dropkan dia dalam atas slide dan kita biarkan incubate selama 2 jam. Barulah kita boleh tengok dalam mikroskop. Okay. So on capsule formation ni, kalau kita buat eksperimen face to face pun saya tak buat benda ni, tapi saya akan bagi information mengenai ni lah. Okay, macam mana kita nak buat capsule formation Place a loop full of Cryptococcus neoformans suspension on the drop of Indian ink on the glass slide. Okay, kita ambil loop full of the Cryptococcus neoformans suspension dan kita letakkan atas glass slide. Tapi kita letak Indian ink dulu atas glass slide kita. Baru kita letak samples. And then kita letak cover slip dan tengok ada tak kapsul dalam tu. Okay. So the yeast Cryptococcus neoformans is usually about 2/3 the diameter of the cells. If the Indian ink is too dark, it may be diluted. So make sure ini ada punya tips okay. Make sure jangan terlalu pekat. Okay. Alright, that's for experiment number 2. Okay. Alright, is there any questions before we continue for experiment number 3? Okay, ada soalan sebelum kita sambung ke eksperimen number 3?
No, sir.
Okay. Tak ada Alright. So quickly we go for the experiment number 3. Okay, so for the further tu awak boleh tengok video macam mana dia buat proses capsule formation tu, awak tengok dekat dalam video kalau ada dan sebagainya. Okay. So we go for the experiment number 3. So what experiment number 3 all about? Okay. So experiment number 3 is more on the filmentous fungi. So filmentous fungi example we use what we call mold. Tadi yeast right? Yeast more on the unicellular fungi. Tapi untuk filmentous, we're using a mold. Okay. So what's the difference between mold and yeast? Okay, number one, mold is the largest microorganism studied in microbiology. Okay. Often, no special staining or biochemical study are necessary to identify the genus since their structure different are often readily visible in microscopically. Sebenarnya kita tak payah pakai apa tu special stain atau sebagainya. Dia mudah dilihat sebab dia paling besar dalam microorganisms. Okay. Though the structural difference, they are most often helpful in the analytical of the filamentous fungi. So fungi mesti ada filamentous, mesti ada hyphae, microhyphae dan sebagainya, kapsul. Okay. So baru kita tahu dia jenis apa fungi tersebut. Dia mesti ada hyphae, microhyphae. Okay. Are different types of spore arrangement and the hyphae. Nampak? Kalau fungi mesti ada merujuk pada special term, iaitu hyphae. Kat bakteria tak ada Okay. But they have what we call spore. So mostly bakteria tak ada spore Several aja dia. Tapi untuk fungi memang ada. Okay. So the main difference is mold usually have the large, light, fluffy and colonial morphology. Dia ada besar. Yang kita nampak macam nipis-nipis. Kalau kat dinding tu nampak macam tokok hitam tu, itu adalah fungi. Dan bila awak sentuh, dia seperti fluffy. Okay. Dia lembut Dia macam sponge. Okay. And colonial morphology. So tapi dalam segi fungi, bila awak sentuh dia malah lembut, tapi bila bukan tengah awak dia jadi hitam Sebab itu adalah dia punya spore. This is the due of its areoles, hyphae and fruiting head. Okay, that's meaning of mold. So how we differentiate it with the yeast? Number one, they have different colonial morphology. Okay, number two, multicellular. Number three, has a special structure and special function. Last one will be their asexual reproduction is a result of pitching off or fragmentation of one of these specialized structure. Okay. So what's the objective for the experiment? Number one, to revise the inoculating techniques of mold onto artificial media. Introduce an alternative method of slide preparation. And then last one, to observe nutrient requirement in fungal growth. Okay. So what are material that we use in the experiment? Okay, number one, equipment. So of course we're using a microscope, sterile scalpel, sterile petri dish with a layer of filter paper, cork borer, buat lubang. Wire loop, same as the bacteria process. Okay, eksperimen bacteria pun kita guna juga. Kita pakai wire loop juga. Cover slip, glass slide, nail varnish, cellophane tips and scissor. Okay, gunting, cellophane tip, okay, pelekat tu. Nail varnish. Okay. What type of culture we're using? Buko, species, Aspergillus, and Penicillium. So mostly kalau awak tengok structure Penicillium, Aspergillus, okay, dia ada macam kapsul kat kepala tu alright. So for staining reagent, we're using 70% of alcohol, okay, for antiseptic. Lactophenol, cotton blue, LPCB or methylene blue, okay, each other Which one Either one you can use for stain, is it methylene blue or LPCB. And last one, sterile distilled water. For what? To letak water drop pada kita punya slide. Okay, media will use PDA or potato dextrose agar, malt extra agar or MEA. Or last one, we corn meal agar, CMA, right? So the one, we have to inoculate our fungi on the artificial media, right? So dip a cork borer on 70% of alcohol, okay? So cork borer tu tempat kita, kalau kau guna tu untuk buat lubang pada kita media, okay? So make sure kita sterilkan dulu, kita gunakan 70% of alcohol. And then flame and air cool. So dekat mana kita gunakan cork borer tu cork borer tu berada di hujung mata dia, alright? Tempat buat lubang tu, dekat ujung tu. Kita kena letak alkohol dan kita flamekan dia, alright? Then bore a hole on the culture plate from any of the mold provide. Okay, so sekarang ni kita dah, mold tu memang dah ada dalam kita punya media. So kita nak buat lubang pada kita punya mold tadi tu. Okay, so bila dah dapat kita punya mold tadi, kita dah buat lubang. So kita transfer the inocular piece of agar from B using a needle onto the center of the A. So sekarang kita dah ada mold. Kita ada mold dalam kita punya culture. So kita pakai cork borer buat lubang. So kita akan ambil lubang yang kita dah buat tu, bulat tu, dekat dalam sampel PDA tu ataupun mold tadi yang sedia ada, kita ambil, kita gunakan needle dan kita transferkan ke arah PDA yang kosong, okay, ataupun sterile petri dish yang ada PDA. Okay. Right, so repeat the same procedure using the flame wire hook. Okay, so kalau kita pakai flame wire hook, touch the colony with a hook and transfer inoculum onto the another PDA plates. Okay, number one, you have using a cork borer. Number two, you pakai flame wire. And then, of course, we have to incubate for 48 hours, which means 2 days or more. Okay. Number two, so tadi adalah inoculate. So number two, macam mana kita nak mounting fungi from the agar media. Prepare a petri dish of agar which approximately 4 mm deep. Okay, maknanya kita letak media dalam kita punya petri dish. Make sure ketinggian kita punya media tu lebih kurang 4 mm. Tengok dari segi tebal ketebalan. With the sterile scalpel, cut agar into 1 cm square. Okay, kita potong agar kita tu. Using the sterile scalpel, transfer the square to the flame glass slide in the glass petri dish containing layers of sterile filter paper. Using the right angle hook, inoculate wire, inoculate the center of each edge of agar square. Immerse a cover slip in 70% alcohol, flame, cool, and place on the agar square. Add the sterile water to the petri dish, incubate at a temperature suitable for the fungi you have inoculated. Okay, and then you have to observe the microscope when the growth have been occurred, usually after 3 days. Okay. Sketch a few representative structure and make appropriate notes. Okay, so sekarang kita boleh observe dia. Remove the cover slip and place on the clean slide. Growth side upward. Place a drop of 70% of the alcohol in the drop of LPCB or methylene blue on the clean slide. Okay. Flick off the agar block into the disinfected jar and mount the growth on the slide of 70% alcohol and LPCB or methylene blue. This mount can be sealed with a nail varnish and will last several years. Okay, so kita nak simpan sampel kita ni, kita pakai nail varnish. Okay. So now we have to use what we call microscopic examination. Using the wet amount of cellophane tapes, make the slides on the given mold. Observe under the microscope. Make a comparison with the method used in the number 2. Okay. So kita ambil sampel kita pakai cellophane tape, kita letak di cellophane tape tu atas kita punya media yang ada mold tadi. So bila awak dapat dah letak cellophane tape, cabut, awak tengok bawah mikroskop. So microscopic examination memang simple. Alright. Number 4, nutrient requirement, work in groups. Okay, ini kalau kita buat face to face. Each group will given 3 plate, which is PDA, MAA, CMA. Okay, so number 1, do as number 1. Number 1 ni kita buat inoculate macam mana. Alright. So make the line cross the petri dish containing artificial media and mark as a dot in the middle of the line. Inoculate your chosen species of mold onto the given media. you can use either a cork borer or a wire hook. Okay, this one untuk kita inoculate sample. Put the inoculum on the dot marks. Incubate at the room temperature. Okay. Measure the diameter of a colony for 24 hours intervals. Continue until the colony cover the whole plate, which will take up to 5 days. Plot the graph of the growth of the fungi, which is the diameter colony, against the incubation periods. Okay. Kita ada buat gambar ajar kat sini. So macam mana kita jalankan proses inoculations dan sebagainya. Okay. Gambar ajar yang pertama, ini adalah proses inoculation. Okay. Kita letak dalam kita punya plates, petak ni. So kita letak filter paper, kita letak kita punya koloni, and then so cover slip kita kena letak 70% alkohol okay, baru kita letak air. Okay, so letak SPCG 70% alkohol sama metal blue, lepas tu letak agar block and seal with the nail varnish. Okay, this is experiment number 3. Okay, so this one untuk eksperimen last tu kita kena plot graph untuk growth of the fungi against incubation periods. Okay. Alright, so is there any question for you for experiment number 2 and 3? Ada soalan tak? So semua video saya akan masukkan dalam future. Okay, ada soalan sebelum saya bagi awak lagi 1 jam untuk memahami eksperimen ni dan plan untuk buat awak punya infographics. Is there any question?